So hi, this is my presentation on cellular respiration and the formation of ATP. So apologize, this might be a little long. So cellular respiration is the metabolic process in which organisms utilize oxygen to break down glucose, which releases energy in the form of ATP, and releasing carbon dioxide and water as waste product. The main steps of cellular respiration include glycolysis, citric acid cycle or Krebs cycle, the electron transport, and oxidative phosphorylation of ADP. So let's look at this step by step. So glycolysis is a catabolic process which breaks down glucose molecules and has basically two stages. No ATP is generated in the first stage, which can be viewed as the trapping and preparation phase. In this phase, glucose is phosphorylazed by ATP to form glucose 6-phosphate. This action is catalyzed by hexokinase. Phosphoglucose isomerase then catalyzes the isomerization of glucose 6-phosphate to fructose 6-phosphate, which is then phosphorylated by ATP into fructose 1,6-bisphosphate through a catalytic action of phosphofructokinase. So ATP is utilized in this stage, 2 molecules of it. So the second stage of glycolysis begins with the cleavage of fructose 1,6-bisphosphate into glyceraldehyde 3-phosphate or GAP and dihydroxyacetone phosphate or DHAP. This action is catalyzed by aldolase. While GAP is directly in the glycolysis pathway, DHAP is not. However, the two are isomers of each other, and triose phosphate isomerase catalyzes the isomerization of DHAP into GAP, which then can be used in the pathway. We now have transformed one glucose into 2 GAP molecules, though have not extracted any energy and actually have used 2 molecules of ATP. The 2 GAP molecules are oxidized in a reaction that is catalyzed by glyceraldehyde 3-phosphate dihydrogenase, transferring a hydride ion from the donor molecule of NAD positive and forming NADH and a hydrogen ion, and 1,3-bisphosphoglycerate. So then phosphoglycerate kinase then catalyzes the transfer of the phosphoryl group in 1,3-BPG to ADP, forming the first ATP and 3-phosphoglycerate. We now have made back the original cost because remember those 2 of those GAP molecules. So that happens with both of those. So the first 2 ATP now plus 2 NADH have been generated. So then phosphoglycerate mutase then catalyzes the rearrangement of 3-phosphoglycerate into 2-phosphoglycerate. Enolase then catalyzes the dehydration of 2-phosphoglycerate into phosphophenylpyruvate or PEP. Pyruvate kinase then catalyzes the transfer of the phosphoryl group from PEP to ADP, which generates another set of ATP and pyruvate. This marks the end of the glycolysis pathway with the generation now of 4 ADP, 2 NADH, and 2 pyruvate molecules. The next step in the cellular respiration is the metabolism of pyruvate into carbon dioxide and water through the citric acid cycle and the electron transport chain. In most tissues, pyruvate is processed aerobically, where pyruvate is oxidatively decarboxylated by the pyruvate dehydrogenase complex to form acetyl coenzyme A, FAD, NADH, and a hydrogen ion. Citrate synthase catalyzes the condensation of, I never say this one right, oxaloacetrate and acetyl coenzyme A to form cityl-CoA, which hydrolyzes into citrate and coenzyme A. Citrate is isomerized in a dehydration and hydration step that is catalyzed by aconitase and forms isocitrate. Isocitrate is then oxidized and decarboxylated by isocitrate dehydrogenase and NAD positive, forming alpha ketoglutarate, CO2, and another NADH. A second oxidative decarboxylation reaction is catalyzed by alpha ketoglutarate dehydrogenase complex, which uses a coenzyme A and an NAD positive to form succinyl coenzyme A, carbon dioxide, and NADH. The thioester of succinyl coenzyme A is cleaved along with the phosphorylation of ADP by succinyl-CoA synthase, forming succinate, CoA, and ATP. Succinate dehydrogenase catalyzes the oxidation of succinate to fumarate, where FAD accepts the hydrogen, which forms FADH2. Fumarase catalyzes the dehydration of fumarate to form L-malate, and malate dehydrogenase catalyzes the oxidization to form, again that word, oxaloacetrate, and NAD in the hydrogen acceptor, forming NADH. So now we have done that whole citric acid cycle as well as the glycolysis cycle. So the electron transport chain, which is a physical link to the citric acid cycle, harnesses the energy released when the electrons flow from NADH and FADH2 to oxygen by a proton gradient across the inner mitochondrial membrane. So all that NADH and FADH2 that was produced during the citric acid cycle can now be used in the electron transport chain to create more ATP. So the electrons flow through three asymmetrically oriented transmembrane complexes, resulting in the pumping of protons out of the mitochondrial matrix and the generation of membrane potential, which facilitates the next. So ATP is synthesized when protons flow back into the matrix. So the components of the electron transport chain are arranged in complexes where the electrons always flow to components with more positive reduction potentials, and so a higher electron affinity. The enzyme complex includes NADH-Q oxidoreductase, succinate Q reductase, Q cytochrome C oxidoreductase, and cytochrome 2 oxidase. So NADH enters the chain at NADH-Q oxidoreductase, or complex 1, where it flows to a series of iron sulfur clusters to ubiquione, or Q, forming Q2, which in turn interacts with the negatively charged amino acid residues. So the flow of 2 electrons from NADH to coenzyme Q through the NADH-Q oxidoreductase leads to the pumping of 4 hydrogen ions out of the matrix of the mitochondrion. Ubiquinol, or Q, is the entry point for the electrons from FADH2, which is after the first proton pumping site, and so leads to less ATP, the forming of less ATP. The flow of electrons from NADH is sufficient to produce 2.5 molecules of ATP and only 1.5 molecules of ATP per molecule of FADH2. Oxidative phosphorylation of ADP is the final step in cellular respiration and the generation of ATP. The flow of protons back into the matrix side through ATP synthase drives ATP synthesis. ATP synthase is a molecular motor consisting of a stationary and a rotating component. The synthesis of a molecule of ATP is driven by the flow of about 3 protons through ATP synthase. In addition, an additional proton is consumed by transporting the ATP from the matrix to the cytoplasm. So in tally, 3 molecules of ATP are consumed in glycolysis and 4 are generated, plus 2 molecules of NADH. In 2 molecules of NADH are formed during the conversion of pyruvate into acetyl coenzyme A to start the citric acid cycle. And then in the citric acid cycle, 2 molecules of ATP are formed, as well as 6 molecules of NADH and 2 molecules of FADH2. So that's the primary role of citric acid cycle, is that production of those NADH. Then during the oxidative phosphorylation of those previous 10 molecules of NADH, they each yield 2.5 molecules of ATP in that cycle, and 2 molecules of the 2 molecules of FADH2 yields 1.5 molecules of ATP each. So that's a total of 26 molecules of ATP formed during the oxidative phosphorylation process. So the net yield of ATP per molecule of glucose is 30. I hope you have a good day, and thank you very much.